Okay, this video is talking about the electrical activity that governs how your heart beats. Your heart is made of two pumps. There's a pump on the right side and there's a pump on the left side, right side and left side. And those pumps need to be coordinated so that they can do effective pumping of blood. When we're monitoring the electrical activity that coordinates this pumping, electro is for electricity, cardio is for heart, and gram is like graph, showing the activity. We can use an ECG or EKG, they mean the same thing, to monitor this. For an ECG or an EKG, we have a stereotypical shape where we have a little bump, we have a quick big bump, and then we have a little bump later. And this repeats over and over and over. This is a standard heartbeat. The differences that we can notice here are labeled. We have this section here, which we call the P wave, this first little bump. This is one, this is one, this is one. These repeat over and over. We call it P, P, Q, R, S, and T. The P is the signal that tells your atria, your right atrium and your left atrium, to contract. They contract at the same time due to a signal from a nerve structure called the sinoatrial node. Sino, like sine wave, and atria, because it's talking about your atria. Node being like a central location. So that electrical signal tells both of your atria to fire at the same time, pumping blood into your ventricles. The next feature is a group of three, the Q, R, and S features. So Q, R, S together are starting here at the atrioventricular node, which is in your right atrium, but controls your left and right ventricles. So the signal goes, I'm going to pick a more visible color here. The signal goes down the septum of your heart. The wall between your two ventricles is called the septum. And it goes down in a whole bunch of nerve fibers that go together. They're bundled up. We call them the bundle of His. This is a name, not like his, but it's just a name of a person. So they're all bundled. They go down the septum, and then they split off into all of the live ends, the ones that are actually delivering the electrical impulse, the shock that tells your heart muscle tissue to contract. Those fibers, those nerve fibers, are called Purkinje fibers. Again, just a name. When they fire, both of your ventricles contract at the same time. When your ventricles are contracting, that's the big squeeze that sends lots of blood out through your aorta or through your pulmonary arteries, your left and your right pulmonary arteries to your lungs. So the arteries up here are receiving high pressure blood pumped by the ventricles, and when they're squeezing, we can measure that high pressure, usually in your arm. It's a convenient place to measure the arterial pressure, and we call that the systolic pressure, or the state is called systole. The last feature is called the T wave, and for that wave, this is a positive thing telling you as clearly as possible, telling your heart as clearly as possible, it's time to relax. So all of the chambers of your heart, both atria and both ventricles, relax during the T wave. The T wave is initiating a state called diastole, or the diastolic pressure, the low pressure if you were measuring your blood pressure. When we measure blood pressure, we measure systolic, which is usually like 120 in a healthy textbook case, whatever, and the diastolic, which is usually 80. 120 over 80 would be like our textbook example of good blood pressure, healthy blood pressure. Of course, that's not true for all individuals, but those are two numbers you'll probably want to remember. And they're measured in a unit called millimeters of mercury. Why millimeters of mercury? That's a good question to ask, but maybe it doesn't fit into this quick video. I'm going to pay attention here to this little chart for a moment. I'm just going to zoom in on it. And there's a few things that I want to point out. One of them is that we have systole. This is the one we measure, ventricular systole. That's what we can actually measure. We can't measure atrial blood pressures at all because the atria are here, and we can't measure the pressure inside of there without sticking a monitor inside the heart, which we want to avoid. So Systole happens for only a very brief portion of a typical heartbeat. The rest of the time, your ventricles are relaxed, and that's diastolic pressure. If we look at the pressure as we go along here, the pressure from your ventricles, or as we would measure in the aorta, is lower, 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 and suddenly your ventricles squeeze and that pressure spikes. This is after the QRS. The QRS is what initiates the big squeeze in your ventricle that raises your blood pressure up to that 120 millimeters of mercury setting. After that, it goes back down to around 80, which is our diastolic pressure. Another thing I want to mention here is we have these two heart sounds. Lub and dub are their stereotypical names. I don't know why. And those are the sounds of two different types of valves closing. The louder sound happens when your atrioventricular valves slam shut. These valves here, they slam shut when your ventricles squeeze. They stop blood from going back the wrong way and only send it the right way. So up to your aorta or out to your lungs. The quieter sound, the dub sound, the second sound, happens when your semilunar valves close. And they don't close as violently, so it's just a less loud sound. So the sounds of your heartbeat that you hear are the sounds of the two types of valves closing, one after the other. Since they happen at the same time, your ventricles both squeeze at the same time and they both relax at the same time, you hear the sound of both your right and your left atrioventricular valves closing at the same time, and your right and your left semilunar valves closing at the same time. So for EKG or ECG, we have these four features, P, Q, R, S, and T. You should know what their features are. You should know the structures in the heart, the sinoatrial node, the atrioventricular node, the bundle of His, and the Purkinje fibers. All of those different structures you'll need to be able to identify the purpose of and find and label in a diagram.